Hi students, today we are going to learn about synaptic transmission. First coming to the definition of the synapse. The synapses are the asymmetric intracellular junctions that mediates rapid point-to-point communication between the neurons and thereby they will form neuronal circuits. So they are important for point-to-point communication between the neurons. Okay, they are intercellular junctions. But synapses not only transfer the information from one neuron to the next neuron, but also processes this information during the transfer. So second point is the processing the information. Next point is the computational properties of synapses differ. So each synapse is different from one another and it cannot be predicted based on their ultrastructure or its location. But the properties of the synapse is based on the synaptic components. Okay. So these are the intercellular junctions between the neurons. They will help in the point-to-point communication and to process the information, and the properties of the synapse are based on the synaptic components. Coming to the characteristic features. This is, you should remember that there is no anatomical continuity, but there is a physiological contiguity. So this contiguity word is important. That means contact is there without connection. So there is a physiological contiguity between the two neurons. Thus, a synapse is a functional junction between the two neurons. So what is a synapse? Synapse is a functional junction between the two neurons. This is the definition. It is most important determinant of CNS function because the information is passed from one neuron to the other neuron via the synapse. What are the functions of the synapse? That is transmission of impulses to and fro from periphery to CNS and CNS to periphery, and they will modify the impulses and it will integrate the impulses or changes the impulses or blocks the impulses. So anything can be happened at the synapse level. Okay, so modification of impulses may be occurring at the synapse level, and higher functions of the CNS like learning and memory are also possible with the help of synapse. So it is important for point to point communication and the transmission of the impulses to and fro, and a modification of the impulses happening at the synapse, and higher functions are also possible at the synapse level. Then coming to the types of synapses. Anatomically, there are three kinds of synapses. They are axodendritic, axosomatic and axoaxonic. Physiologically also, we have three types of synapses. They are chemical synapse, electrical and conjoined synapse. First coming to anatomical classification, this is the axoaxonic synapse. So, one axon is communicating with the another axon. Okay. So, these axoaxonic synapses are least common, seen in the spinal cord. Here, second one is axodendritic synapse. So, axon is communicating with the dendrite of the another neuron. This is the most common type 1. This is the axodendritic synapse. Type 1 is the most common. Motor neurons of the spinal cord and excitatory synapses in the cerebral cortex and climbing fibers of the cerebellum will exit these synapses. So, axodendritic synapse is the most common synapse. Next comes the axosomatic synapse. Axon is communicating with the soma of the another neuron. It is called type 2 synapse. It is less common. It is shown by some motor neurons of the spinal cord, basket cells of the cerebellum and autonomic ganglia. So, axodendritic synapse is the most common. Then, axosomatic synapse is the less common. And axonic is the least common. And rarest is the dendrodendritic synapse, seen in the olfactory bulb between the mitral cell and granule cell. We will see some aspects of formation of the synapse. Okay, how a synapse is formed, how it is survived, and how some synapses are eliminated, but not in detail. So some important points only we will touch. So a synapse is formed. First, a neuron is formed. So the growing neuron, it should have the path finding. So where to go, and it should synapse with another neuron. So to which neuron it should synapse, that is target recognition. So axon should find its path, that is axon path finding, and it should synapse with the target, that is the target recognition. These two are important for a synapse to be formed. So this axon path finding is with the help of some guidance forces. So these forces are produced by guidance molecules. These guidance molecules are the proteins that help with the axonal guidance. And target recognition, how axon know that they have to go to this particular target only. This is the target recognition. See this, Raman Y. Kajal, he first described the growth cones and observed that they move in a circuitous route towards their targets. So he is the scientist found growth cones. So an axon is growing. So at the end, that is where the end part where the axon is growing is described as growth cone. And he described that growth cones and observed that they will move in a route towards their targets. So this is the anatomy of a growth cone. So it is having microtubules and actin filaments, and it is throwing phylopodium. Okay, these are the finger-like structures, phylopodium, and in between you have lamellipodium. So it is enriched with the actin filaments, okay, and microtubules. So this is a growth cone. Next, coming to the guidance forces. Guidance forces is because of the guide post cells, okay, and pre-existing new axons. So guidance forces are generated by guide post cells and pre-existing axons. These will affect the outgrowthing of the developing axons. In addition to these influences, axons will also receive long-range guidance information. So an axon can get short-range or long-range guidance information, and this information can be attractive or repulsive. So it helps to push or pull the growth cone in the correct direction towards its target. So an axon will get both short range and long range guidance information, and this information can be attractive or repulsive. Based on the information, it will be growing in that direction, or it may go away from that direction. So short range guidance mechanisms like interaction with the guide post cells. So what happens? Guide post cells are present at some intervals of a distance. So these axons will go towards the guide post cells, and some axons will form bundles in the peripheral nervous system, and they will form fasciculations and involve contact mediated detection by the growth cone phylopodia. So this growth cone has phylopodia finger-like structures. It will recognize the guide post cells. So these contact may be attractive or repulsive. And long-range diffusible factors may also influence the axonal guidance, both by attraction and repulsion. It is likely that axons are affected by both local and long-range forces acting simultaneously. So what you have to understand is local, that is short range guidance, and long range guidance. These forces are acting simultaneously. Local or short range guidance is because of identification of guide post cells by phylopodia, and long range forces is because of some diffusible factors. See this is the cell body, and this is the growing part, this is the growth cone. So it is coming towards this neuron. And you can see attractive forces or repulsive forces. If there is attractive force here, the neuron will go there. And if there is repulsive force here, the neuron won't go there. So attractive force is produced by some diffusible factors serve to direct axons to their appropriate targets. So if a long range diffusible factor is attracting, so the neuron will go to that side. Repulsive signals in the form of concentration gradients of soluble proteins helps to direct the axons away from the incorrect destinations. So if a repulsive force is there, then the axon won't go there. See here, the guidepost cells, here they are present, here they are present. So axon will come towards the guidepost cells. Guidepost cells act as intermediate targets and helps to guide the growth cones to reach their final target. And many axons will form a bundle known as axon fasciculation. So this is the pioneer axon, which is blue in color, serves as a scaffold for the outgrowth of the new axon from another cell, that is a purple cell. So this cell has formed an axon and this will form a platform so that this cell body will uh, form a new axon and connect this axon to form a fasciculus. Now target recognition. So how this axon is recognizing the target. So the Rogers-Perry scientist, he gave the chemoaffinity hypothesis. Okay, so he suggested that the positional information is encoded in the form of gradients of signaling molecules at the target, which would be detected by complementary gradients of receptors on the axons. So what he is telling, the axons, they are having receptors, that is the phylopodia, all these are having receptors, and the signaling molecules are there far away. So these receptors will go to that particular signaling molecules. That is called chemoaffinity. This model implies that the positional information can be encoded by a small number of molecules and that this information can be read by axons as they maneuver in the target region. The chemotropic guidance of axons by diffusible factors that are secreted by cells can involve attractive or repulsive forces. So chemoaffinity hypothesis by Rogers-Sperry, he told that the positional information where the axon should go, it is given by the some molecules which are soluble and diffusible factors. It is called a chemotropic guidance. So with the help of this guidance, the axons are going to that particular target, and that particular target is having that signaling molecules. Now, one more important thing is the molecular logic of the synapse formation and the involvement of SAMs. SAMs means synaptic organizing molecules. These are the principal agents in organizing synaptic junctions. How? By engaging transcellular interactions, these SAMs are thought to nucleate nascent synapses, drive synapse maturation, and controls the properties of the synapse and regulate the synapse elimination. So these SAM molecules are important for all these functions which are occurring at the synapse level. So SAMs will perform these actions by signaling in both directions, that is pre to post and post to presynaptic direction. So no single SAM is identified, but an orchestra of SAMs are needed to do all these actions. So to do these diverse synaptic junctions. So many types of synaptic junctions are there and many properties are there. All these are because of SAMs, synaptic organizing molecules. See this? This is the nascent synapse. This is the mature synapse. And this is the eliminated synapse. So in nascent synapse, what is happening? This is the nascent synapse. So while neurogenesis and neuronal growth, neuronal migration is occurring, then axon will be guided. Axon guidance and dendritic organization happen. So first what happens? Neurogenesis is happened. Then neuronal migration is happened. Then axon is grown and dendritic has grown. Then synapse is formed. That is the nascent synapse here. So it will engage the transcellular SAM complexes, and it will perform pre and post synaptic intracellular signals, for example via cyclic AMP pathway, and it leads to initial assembly of the synapse. So this is the presynaptic specialization. It is like this. Okay. So this is a post-synaptic specialization area. So it is like this. So this is a nascent synapse. So our SAM complexes, they will act pre and post synaptically. So they will form pre and post synaptic intracellular signals. So they, for example, they are acting by cyclic AMP pathway, and they are causing initial synapse assembly. Then how mature synapse is formed? This is a mature synapse. Okay. So it is having neurotransmitter receptors at the active zones, and adhesion protein complexes are formed, and here exocytosis is also happening. So signaling via diverse SAM complexes. So many SAM complexes will be attached, and many will be sending signaling mechanisms, and there is activation of the CASK pathway. Okay, pre and post synaptic protein activation, that is activation of the CASK protein occurs, and this leads to specification of the synapse properties. So synapse properties, they are depending on the SAM complexes. Then some synapses are eliminated. So see this disassembly of SAM complexes. If the SAM complexes go away, then what happens? Pre and post synaptic disruption of the signals occurs. So there is no signaling pathway occurs in the pre and post synaptic region. It leads to elimination of the synapse. So our SAM complexes is helpful for initial synapse assembly and for specification of the synapse properties through various pathways. SAMs perform two overlapping functions. What are they? They will organize the synapses. They will organize the synapses. That is, they will make the synapses, and they will specify the synapse properties. That is, shaping the synapses. So they will make the synapse and they will shape the synapse. So main important were neurexins and these PT series ones. So these dotted lines are cis interactions, and these continuous lines are trans interactions. So this is the presynaptic membrane, this is the synaptic cleft, and this is the postsynaptic membrane. So these are interacting. The presynaptic and postsynaptic proteins are interacting. So all these are SAMs. Most important ones are neurexins and PT series. Next, coming to the types of synapse, we know that we have chemical and electrical synapse. So in electrical synapse, what is there? Anatomical continuity is there. So there are gap junction channels through which smaller ions and molecules can be passed. Here we have chemical synapse, that is a chemical should be released here. Okay, and the chemical should act on the postsynaptic membrane in inotropic or metabotropic receptor. So see the differences between the electrical synapse and chemical synapse. Electrical synapse, the speed of conduction is really fast. Almost there is no delay. And here in the chemical synapse, it is having a delay of approximately 1.5 milliseconds or more. So the speed is very much slow, and maximum speed it can achieve is 0.3 milliseconds. But typically 1.5 milliseconds or more. So what he is saying is the speed is 1.5 milliseconds to 0.3 milliseconds. And there is a synaptic delay is present. Why synaptic delay is present? Because the neurotransmitter has to be released and it should act on the postsynaptic receptors and it should open. Then ion should come, and there should be formation of end plate potential. After that, action potential should be generated. So here it is not there. There is simply the ions will be coming from presynaptic presynaptic junction to the postsynaptic junction. intermembranous space, that is the synaptic left. The gap junctions physically connect the presynaptic and postsynaptic membrane. So only 4 nanometer synaptic left space is present. Here in the chemical synapse, synaptic left is physically separated by approximately 20 to 40 nanometers distance. Signaling messengers are ions or charged particles. They will send the electrical currents. Here the chemical synapse requires chemical signal, that is chemical messengers. That is neurotransmitters are required. Here the intermediates in transmission, there is no intermediates in the transmission. Here the chemical messengers that are transported across the intermembrane space inside a synaptic vesicle. So there is a transmitter, that is the neurotransmitter is present in the 30 to 50 nanometer sized synaptic vesicles. Coming to direction, this electrical synapse is bidirectional. From here to here, the ions can move. Chemical synapse is typically unidirectional. So from here to here it will come, because neurotransmitter present here and receptors are present here. So what is the synaptic knob or synaptic button? As the axon approaches the synapse, it will lose the myelin sheath. If it is myelinated axon also, at the synapse level, it will lose the myelin sheath, and it divides into number of fine branches. These fine branches will end in small swellings called synaptic knobs or synaptic buttons. They will make a synapse with a soma or dendrite or on the neuron also. Okay. Each synaptic knob, what it is consisting of? It is consisting of many mitochondria and synaptic vesicles which is having neurotransmitter. So why this mitochondria is important? Mitochondria provide ATP which is required for the synthesis of the neurotransmitter. So actually we have two types of neurotransmitters mainly. So first one is the peptide neurotransmitter. So it will be synthesized in the nerve cell body and transported across the microtubules to the nerve terminal. But the non-peptide, for example, acetylcholine neurotransmitter, it will be synthesized at the synapse level only. That is at the nerve terminal. That is at the synaptic buttons. So that will require ATP. So mitochondria is providing ATP for the synthesis of neurotransmitters. We can see three types of synaptic vesicles. Small clear vesicles have acetylcholine, glycine or GABA. And small vesicles with a dense core, they will have catecholamines. And the large vesicles with a dense core, they will have neuropeptides as a transmitters. Okay. So small clear vesicles have acetylcholine, glycine or GABA. Small vesicles with a dense core, they will have catecholamines. Large vesicles with dense core, they will have neuropeptides. Okay. Then coming to the presynaptic membrane. This is the axonal membrane lining the synaptic knobs. So on the inner side of the presynaptic membrane, there are dense zones of cytoplasm. Okay. So it will form a presynaptic vesicular grid for organization of the channeling of the vesicles to the presynaptic membrane. So presynaptic membrane is having dense cytoplasm, zone of dense cytoplasm, that is forming presynaptic vesicular grid. So it will organize the channeling of the vesicles, that is the vesicles should go through that channels. Then coming to the synaptic left. It is a small gap between the pre and postsynaptic membranes, approximately 20 to 40 nanometers wide, and it is filled with the extracellular fluid. Okay. And having some glycoproteins also. Neurotransmitter initially released into this synaptic left. Okay. So enzymes are present in the synaptic left. They will destroy some amount of neurotransmitter. Then coming to the postsynaptic membrane. This is the Axonal membrane lining the postsynaptic area. Here also we have dense cytoplasm on the inner aspect. So this is the zone of dense cytoplasm. This is known as active zone of the synapse. So active zone is present on the postsynaptic membrane. So this is containing large number of receptor proteins, okay, which will protrude to the synaptic cleft. So neurotransmitter, when it is released into the synaptic cleft, it will bind to the receptor on the postsynaptic membrane. So the receptor proteins. So they are proteins, receptors actually. They are of two types. It may be ion channel receptor proteins. That means when the neurotransmitter has binded to that particular receptor, the ions will enter or they will go out of the cell. That is sodium, potassium chloride, etc. And that is inotropic receptors. Second is the metabotropic receptor. That is enzymatic type of receptor proteins. So when they will when the neurotransmitter attaches to this receptor, what happens? Series of changes occurring. So it will cause a series of changes in the cell, and it will lead to cellular gene modification, okay, and uh, and some synaptic modulator effect can also be produced with the help of enzymatic type of receptors. Next, coming to the, so we can see this. This is the axon terminal. Here we have synaptic vesicles. So from here to here the signal is going. This is the chemical synapse. This is the electrical synapse. We have gap junctions. These gap junctions are made up of proteins called connexins. The direction of signaling may be here to here or here to here. That is not shown here. Next, coming to the steps or a process of chemical synaptic transmission. So what are the steps of transmission of the at the synaptic level? First one is the release of neurotransmitter. Then development of EPSP and IPSP. Then activation of neurotransmitter from the synapse. Inactivation of the NT from the synaptic cleft, and development of action potential. So development of action potential will be discussed in detail in the chapter of the nerve. So you can see the video on the nerve. So this is we are not discussing. Just the ultimate function is to develop the action potential only. So what are these three steps? First, coming to the release of neurotransmitter. So this neurotransmitter is present in the presynaptic vesicles. So the neurotransmitters are divided into low molecular weight transmitters like acetylcholine, glycine, and glutamate, okay, and neuropeptides. Over 40 are identified. And there are two types of secretory vesicles for this. So we have seen actually three types of secretory vesicles. So they are small clear vesicles having acetylcholine, glycine, and gamma. Small vesicles with dense core containing catecholamines, and a large vesicle with dense core containing neuropeptides. So small chemical neurotransmitter vesicles we are seeing here like acetylcholine. So it is responsible for fast synaptic signaling. Okay. Example, all these things. So enough vesicles are present at the nerve terminal. The exocytosis is occurring because of the increase in the calcium concentration in the cytoplasm of the presynaptic membrane. You see this? The vesicle, this is the vesicle. So the vesicles is having some transporters. So these red dots are actual our neurotransmitters. For example, acetylcholine. So here we have H plus neurotransmitter antiporter, and this is a H plus ion. So this H plus ion is entering into this. This is a vesicle, and it is utilizing ATP. It is hydrolyzing ATP to ADP and PI. This ATP is produced from mitochondria. Now, this is a H plus neurotransmitter antiporter. So this name is called V-type ATPase, that is vesicular type ATPase. It is causing ATP hydrolysis and allowing H plus ion. V-type ATPase, vesicular ATPase. And this is the H plus neurotransmitter antipotent. So it is throwing the H plus ions out and taking the neurotransmitter inside. That is the antipotent, anti-direction. So this step is the first step. That is the import of neurotransmitter into the vesicle. Now coming to second step. This vesicle will move to the active zone. So active zones are nothing but they are the zones of dense cytoplasm in the pre and post synaptic membrane. Now this will move to the active zone. Then they will dock at the plasma zone. See this? In step 2, the vesicle moves to the active zone near the plasma membrane and dock at a defined membrane sites by interacting with the specific proteins. So here you have presynaptic plasma membrane. It is docking at the plasma at some zones. Then calcium is entering into the cell. Then what happens? A rise in cytosolic calcium triggers the fusion of this vesicle, docked vesicle, and release of neurotransmitters occurs. So this process involves snare proteins. It will be discussed in detail later. In the synaptic vesicle membrane proteins, these synaptic vesicle membrane proteins, then they are recovered by endocytosis. Okay, recovered by endocytosis. How? That is we have many types of endocytosis. Here it is using clathrin coated endocytosis. So the clathrin coat is also then depolymerized and it will release the actual vesicle. Okay. So here we have regulated exocytosis regulated by calcium, and here you have clathrin mediated endocytosis. Here dynamine is a protein, it will cut here and the entire vesicle will move away, and clathrin is also depolymerized and the vesicle is resynthesized. See here, snare hypothesis. So how a vesicle is exocytosed. So how the contents of the vesicle is exocytosed. It is because of the snare proteins. So see here, we have, this is the vesicle, this is the plasma membrane, that is the presynaptic membrane. V snare means the snare proteins which are present on the vesicle. What are this? What are those? Those are synaptotagmin, okay, synaptotagmin and synaptobremin. And here T snares, that is target membrane snares, they are having syntaxin and SNAP25. This is a calcium channel, this is neurexin. So this calcium and neurexin are helpful for the stabilization of this snare proteins. This synaptotagmin is a calcium sensor. So when calcium enters into it, calcium will bind to the synaptotagmin. See this? Some toxins, that is those are the toxins released by clostridial members, that is tetanus toxin and botulinum toxin. So we have many types of botulinum toxin. For example, botulinum A, C and E, they will cleave the SNAP25. So they will cause cleavage of these peptides. SNAP25 is cleaved by C, A and E type of botulinum toxin. And botulinum C is affecting syntaxin. So botulinum C is affecting syntaxin and SNAP25. And botulinum B, G, D, F, they are attacking on synaptobremin. So they are attacking on vesicle snare proteins. So these are not allowing the vesicle to fuse here and the vesicle cannot be exocytosed. Okay. So here we can see tetanus toxin. This is also affecting on this blue colored one, that is synaptobremin. So here only. So botulinum G, B, D, F and tetanus toxin is acting on the synaptobrevin. C is acting on syntaxin and SNAP25. And SNAP25 is also affected by botulinum A and E along with C. So, then what happens? Development of EPSP and IPSP. So, now the vesicle has exocytosis. That is, when the vesicle is docked and when it is released because of the help of snare protein. So, this synaptobrevin, it will combine with the SNAP25 and syntaxin, and this will lead to the fusion of this vesicle membrane with the presynaptic membrane, and it leads to the exocytosis. Then what happens? The in the postsynaptic membrane, there will be development of excitatory or inhibitory postsynaptic potential. So if it is releasing positive ions, then it is causing excitatory postsynaptic potential. That is, if it is releasing sodium into the cell, then it will lead to EPSP. If it is releasing potassium, that is, potassium is going out or chlorine is coming in, then it leads to IPSP. That is, membrane potential is further going into negative side in IPSP. Here the membrane potential is going towards the positive side in EPSP. So see the difference between EPSP and IPSP. EPSP is an electrical change on the postsynaptic membrane which is caused by excitatory neurotransmitters. It is caused by IPSP is caused by inhibitory neurotransmitters. Okay. So EPSP is leading to depolarization. IPSP leads to hyperpolarization. So these are the some examples. So these are some differences. Then these EPSP and IPSPs. EPSP itself can summate or IPSP itself can summate, and EPSP and IPSP can also be summated both in temporally and spatially. So what is meant by temporal summation? So two excitatory stimuli, they will be close in time to cause EPSPs to add together. What is spatial summation? Two simultaneous stimuli will be coming from different locations causing EPSPs that will add together. So EPSPs can add together, IPSPs can add together. EPSP and IPSP can also add together, and they leads to cancellation of the each other's effect. Next, coming to the removal of the neurotransmitter. That is how the inactivation of the neurotransmitter is happened. It can be broken down by enzymes. For example, acetylcholinesterase breaks down the acetylcholine in the synaptic left. So this enzyme is present in the synaptic left, and on the postsynaptic membrane, it will cleave the acetylcholine in a two-step process. And many nerve gases, okay, and insecticides work by blocking the acetylcholinesterase, and they will prolong the synaptic communication. Second one is the recycled by uptake. So most neurotransmitters are removed by sodium neurotransmitter symporter. That is, the presynaptic membrane will take up the neurotransmitter along with sodium symporter. Okay? And this neurotransmitter will be inserted into the vesicle. And some neurotransmitter will be diffused. It is simple diffusion away from the site. So these three can be happen to the neurotransmitter. First one is broken down by enzyme, or it may be uptaken by presynaptic membrane for recycling process, or it may go in a simple diffusion. Next, development of action potential. It is because of the synaptic integration, that is summation of EPSPs, summation of postsynaptic potentials. This we have already seen. And generation of initial segment spike. This we will see. And generation of propagated signals, that is the generation of action potential. See, this is the spike potential. So what is spike potential? This, for example, you are stimulating the dendrite of a neuron. Then what happens? This EPSPs are summated. Then it is leading to the spike potential. So that is, this spike potential is initiated at the initial segment. That is the starting segment of the axon. So, if there is a depolarization of the 8 to 10 millivolts also at the level of the initial segment because of the high concentration of the sodium and potassium ion channels at the axon initial segment, there will be opening of the voltage gated sodium channels, and it is leading to the spike potential. So it is leading to depolarization at the initial segment, and there is generation of the action potential, and it will be propagated down the axon. And there is exists some plasticity of the initial segment. Always it is not at the just starting of the initial axon only. So, and the length is also variable. So for example, for some axons we have 5 to 100 microns initial segment, axon initial segment, and for some it is somewhat away distance, okay, from the soma, and in some there is a long, there is much more distance from the soma, okay, like that. And in some, and in unmyelinated neurons there is no axon initial segment, okay. So it may mostly the position of the axon initial segment is differing, so it is based on the excitability of that neuron. So the excitability of the neuron is dependent on the position, okay, that is site of the axon initial segment and on its length also. Then coming to the role of ankyrin G in the axon initial segment. So what is this ankyrin G? It is a protein. It is a protein. So what is the here it is the ankyrin G protein. Okay. So see here, this is the beta spectrin, this is the alpha 2 spectrin. So this is the beta 4 spectrin, this is the alpha 2 spectrin. So these four join together, and these are the ion channels. These are the CAMs, that is cell adhesion molecules. This is the plasma membrane, and this is the actin filaments. So what happens? Our ankyrin G protein will hold the alpha 2 spectrin, beta 4 spectrin, and ion channels and CAMs with the actin filaments. So ankyrin G recruits and anchors the ion channels, mainly sodium ion channels voltage gated 1.6. So we have many different types of channels, and potassium voltage gated channel 7.2 and 7.3 are holded by ankyrin G molecules. It will hold via several binding sites. Okay, several binding sites are present on the ankyrin G molecules. It is a membrane binding domain. Okay, it is a membrane binding domain, and see here, it is a membrane binding domain. It is a spectrin binding domain, and this is a tail for the ankyrin G. So ankyrin G binds to an interaction motif with the second and third intracellular loops of sodium voltage gated sodium channels. So voltage gated sodium channels, they are having many intracellular loops. So it will bind to second and third intracellular loops of the sodium channel. So this results in a 30-fold increase in the concentration of the voltage gated sodium channels at the axon initial segments when we compare the number to the dendrites or the distal axon. And this voltage gated sodium channels are primarily responsible for the initiation of the action potentials at the axon initial segment. Now coming to the inhibition at the synapses. So we have some inhibition at the level of synapse. These are four types. What are they? Postsynaptic inhibition, presynaptic inhibition, feedback and feed-forward inhibition. So coming to the postsynaptic inhibition, it is a direct inhibition. That is, it is due to development of IPSP because of the inhibitory neurotransmitter. So, for example, when inhibitory neurotransmitter is released, then what happened? Chlorine is entering into the cell or potassium is going out. So, it is leading to the inhibition at the level of synapse. That is called postsynaptic inhibition or direct inhibition. So, there is no production of action potential in the postsynaptic membrane. Okay. Sometimes it may also occur due to refractory period also. Next coming to presynaptic inhibition. So here what is this? It is the indirect inhibition. That means it is due to release of an inhibitory substance onto the outsides of the presynaptic nerve fibers. So we have two nerve fibers, presynaptic and postsynaptic. This presynaptic nerve is inhibited by another neuron by releasing inhibitory neurotransmitters. Okay, so that is the presynaptic inhibition. That is presynaptic neuron is inhibited. Indirect inhibition. Presynaptic inhibition occurs in many sensory pathways in the nervous system. For example, gating of the pain transmission. So, control of the pain occurs because of this presynaptic inhibition. And some adjacent sensory nerve fibers also, they will mutually inhibit one another. So, it will minimize the sideways spread and mixing of signals in sensory tracts. So, how this presynaptic inhibition is helpful? It is helpful for the cutting down of the sideways spread of the stimulus, and it will cut down the mixing of the signals in the sensory tracts. Okay, so see here, this is a one neuron, this is another neuron, this A neuron, this is B neuron. This A neuron is inhibited from the C neuron. So this is the presynaptic inhibition. So if this neuron is causing release of the uh, entering of the chloride or releasing of the potassium here, and this is called postsynaptic inhibition. So in postsynaptic inhibition only two neurons will be present. In presynaptic three neurons will be present. Next, coming to the feedback inhibition or Renshaw cell inhibition. So here you can see in the spinal cord what is happening. The neurons inhibit themselves in a negative feedback manner. So here you can see upper motor neuron is coming down and it is stimulating the lower motor neuron, that is alpha motor neuron, A alpha. This alpha motor neuron is giving a collateral and it is stimulating the Renshaw cell neuron, which is in the spinal gray matter. This Renshaw cell neuron is inhibiting the alpha motor neuron, that is A alpha motor neuron. This is called feedback inhibition. Okay, so it is seen in the spinal cord motor neurons like alpha motor neuron, which is situated in the anterior gray horn. Okay, then coming to the feed forward inhibition. In cerebellum, we can see between the basket cells and the Purkinje cells. So what is happening here? Feed forward inhibition. Actually, what is happening? This Purkinje cell is sending its impulses to the deep cerebellar nuclei. Now this is inhibited by basket cells. How? These are the parallel fibers, that is the output from the granule cells. So this is discussed in detail in the uh, cerebellar videos. So this is the output from the granule cell which are forming parallel fibers. Now these are exciting both Purkinje cells and basket cells. So before this Purkinje cell shoots, this basket cell is inhibiting the Purkinje cell. This is called feed forward inhibition. So the discharge from the Purkinje cell is reduced and output from the Purkinje cell is controlled. What is the significance of the synapse inhibition? So we have learned about the four types of synapse inhibition. What is the significance? So it offers restriction over the neurons and muscles. So the excess stimuli are inhibited and various movements can be performed properly and accurately. So excess movements are cut down. And this inhibition helps to select the exact number of impulses and it will block the excess ones. Okay. Poisons like strychnine destroys all the inhibitory functions. So it is causing convulsions. Okay. So it is inhibiting the postsynaptic inhibition. So there is no inhibition. It leads to convulsions and leading to strychnine poisoning, some poisoning. And in Parkinsonism also, inhibitory system is impaired and it is leading to the motor disorder. So this is the significance of synapse inhibition. So up to now what we have learned, the synapse, the definition of the synapse and the anatomical and physiological types of synapses. Anatomically it is divided into axodendritic, axosomatic, axonic, dendritic synapse. Physiologically chemical, electrical and conjoint synapse. Okay. And chemical synapse, we have discussed the structure of the chemical synapse in detail, that is It is having presynaptic membrane, synaptic cleft, postsynaptic membrane. Okay, and synaptic knobs and buttons. And we have seen the steps of a chemical transmission. That is release of neurotransmitter and development of EPSP, IPSP and its summation. Then we have seen the development of action potential and inactivation of the neurotransmitter from the synaptic cleft. Okay. Then we came to the inhibition of the synapses. That is postsynaptic inhibition or direct inhibition, presynaptic or indirect inhibition, feedback and feed forward inhibition. Then we will go to properties. All these are the properties of synaptic transmission. So first one is the one way of conduction. Second one is the synaptic delay. Next, synaptic fatigue. Then convergence and divergence. Then summation. Then occlusion phenomenon. Then subliminal fringe effect. Then facilitation, reverberation, reciprocal inhibition, after discharge, effect of acidosis and hypoxia, effect of drugs, and synaptic plasticity and learning. All these are the properties at the synaptic transmission. First, coming to the one-way conduction. The chemical synapse allows only one way of conduction, that is from pre to postsynaptic neuron, and never in the opposite direction. This is called Bell-Megendi law. Because the neurotransmitter is present at the presynaptic area and receptors present on the postsynaptic area. Hence, antidromically conductor signals conductor signal dies out at soma due to lack of chemical substance. Significance is for orderly neural function. So one way of conduction is important for orderly functioning of the neurons. Next, coming to the synaptic delay. So during the transmission of a signal, there is a delay. Why this delay is occurring? Because of the all these steps should happen in between the presynaptic release of uh, neurotransmitter and the generation of action potential in the postsynaptic cell. That is, discharge of transmitter should be occurring, then diffusion should occur, then this transmitter should act on the receptor, the receptor to increase the membrane permeability, then sodium should diffuse in and it should cause a EPSP and this should lead to action potential. So all these steps will require some time. So there is a delay at the synapse, that is called synaptic delay. So what is the significance of this delay? That is, conduction along a chain of neurons will be slowed down if there are many synapses, and it is sometimes possible to know the reflex pathway, whether it is monosynaptic or polysynaptic pathway, by measuring the delay. That is how many synapses are present between the one reflex we can roughly calculate. Next property is the summation property of the synapse. So it also has spatial and temporal summation. What is meant by spatial summation? When many presynaptic terminals are stimulated simultaneously, what happens? There is a summation effect at the postsynaptic neuron. And when there is a stimulation of the presynaptic terminal repeatedly, okay, then there occurs temporal summation in the postsynaptic neuron, and these summations will cause increase in the EPSP, which ultimately leads to action potential. Then coming to subliminal fringe effect. Okay, sorry. Then coming to occlusion phenomenon. So what is the occlusion phenomenon? So you see this ABC neurons, they are stimulating XYZ neurons. So because A is stimulating X and B is stimulating X and Y, C is stimulating Y and Z. Then what is happening? If the response to stimulation of the B and C together leads to some excitation of the XYZ neurons. Okay. But if you stimulate ABC also, then also there is stimulation of the XYZ neurons only. Why it is happening? Because B is supplying to X and Y and C is supplying Y and Z. So this Y is getting its input from the B and C, and X is getting input from the A and B. So if you stimulate either this or this neuron, it is getting stimulated. And if you stimulate if you want to stimulate this, either this or this can be stimulated. So the response to stimulation of the B and C together is not as great as the sum of the responses to stimulation of the B and C separately. So if you stimulate B, X and Y, that is two neurons got stimulated. If you stimulate C, Y and Z are stimulated. So 2 from the B, 2 from the C. But if you stimulate B and C together, only 3 neurons are stimulated. Okay. So this is a this decrease in the expected response. Okay. So what we thought, B is stimulating 2 neurons and C is stimulating 2 neurons. So if you stimulate B and C, we can stimulate 4 neurons, but only 3 neurons have stimulated. So this is a decrease in the expected response. This is due to presynaptic fibers sharing postsynaptic neurons. This is called occlusion. Okay. So this is the 2 plus 2 is not equal to 4 here. It is equal to 3 only. Okay. Next, coming to the opposite word, that is subliminal fringe effect. That is here 2 plus 2 is equal to 5. How it is happening? But if you take more neurons, what is happening? Because of the firing of many neurons, this facilitated zone also coming into discharge zone. Suppose if two neurons are, this one neuron is stimulating some 5 neurons and the second neuron is stimulating some 5 neurons. When they are stimulated together, they are stimulating 15 neurons, not 10. So that is because these facilitated zone neurons are coming in the discharge zone because they are stimulated by simultaneously two nerve fibers. This is the subliminal fringe effect. Next, coming to the synaptic plasticity. Plasticity refers to the capability of being easily molded or changed. So that is called a plasticity. The synaptic transmission can easily be increased or decreased on the basis of past experience. So these are the 5 forms of synaptic plasticity. What are they? Post-tetanic potentiation, long-term potentiation, long-term depression, habituation and sensitization. See, post-tetanic potentiation. If you apply tetanizing stimuli in presynaptic neuron, what is caused? It results in increase of the postsynaptic potentials, which only lasts for some minutes to hours. Why it is occurring? Because of increased calcium influx in the presynaptic neuron, which increases the release of neurotransmitters. Okay, that is the post-tetanic potentiation. Next, coming to the long-term potentiation. Okay, it occurs in the hippocampus. So, if a post-tetanic potentiation gets more prolonged and lasts for some days, it is called long-term potentiation. It occurs due to increase in intracellular calcium in the postsynaptic neuron rather than presynaptic. So, post-tetanic potentiation, calcium is increased in the presynaptic neuron. In long-term potentiation, calcium is increased in the postsynaptic neuron. For example, it occurs in the hippocampus. Next, long-term depression. It is opposite of long-term potentiation. It also occurs in the hippocampus and the cerebellum. So there is a slower stimulation of the presynaptic neurons with a decrease in synaptic conduction following decreased calcium influx. So that is depressed long term. Okay, long term that neuron is a depressed presynaptic neuron. This leads to long-term depression seen in hippocampus and cerebellum. Next, coming to the habituation. When a stimulus is benign and is repeated over and over, the response to the stimulus gradually disappears. Okay, that is called habituation. That is habituated. This is associated with the decreased release of neurotransmitter from presynaptic terminal because of decreased intracellular calcium. The decreased calcium is due to gradual inactivation of the calcium channels. Okay, so habituation, if you give a benign stimulus repeatedly over and over, so the response for that particular stimulus is gradually disappear. This is called habituation. This is because of the decrease in the calcium intracellular in the presynaptic neuron because of the inactivation of the calcium channels. It can also be short term or it can also be long term. Next, coming to sensitization. Okay, so it is the opposite of habituation. So there is a prolonged occurrence of increased postsynaptic responses after a stimulus is paired once or several times with a noxious stimulus. So sensitization is occurring after a noxious stimulus is given. It is because of calcium mediated changes. and adrenal cyclase that results in greater production of cyclic AMP. So, if you give benign stimulus over a long period, it leads to habituation. If you give a noxious stimulus, it will lead to sensitization. And all these are mediated by calcium. And a facilitation. When a presynaptic neuron is stimulated with several successive individual stimuli, each stimulus may evoke a larger postsynaptic potential than that evoked by the previous stimulus. This is called a facilitation. Next, coming to the fatigue. Fatigue of synaptic transmission. So when excitatory synapses are repeatedly stimulated at a rapid rate, what happens? There is a fatigue of synaptic transmission. Why it is occurring? Because there is a particular amount of neurotransmitter only. That is, the terminals contain they can store some excitatory transmitter only to about 10,000 action potentials. If you give more than that, the transmitter is exhausted and it is not synthesized in that minimal period. So there is exhaustion of the neurotransmitter. So there is a fatigue at the synaptic level. It is a protective phenomena. Okay. Next, effect of acidosis or alkalosis. Alkalosis greatly increases the neuronal excitability. That is, if arterial blood pH it goes from 7.4 that is normal to 7.8 like that, this causes cerebral epileptic seizure. Why? Because this is causing alkalosis, it increases neuronal excitability. Okay? And this can be demonstrated especially by asking a person who is predisposed to epileptic seizures to overbreathe. So, if a person overbreathes, an epileptic person, if overbreathes, what happens? Alkalosis is developed and it is leading to seizures. See here, overbreathing blows off carbon dioxide, so there is elevated pH in the blood momentarily. It will precipitate epileptic attack. And acidosis, exactly opposite, greatly depresses neuronal activity. Okay. Next, effect of hypoxia. Neuronal excitability is also highly dependent on the oxygen. So cessation of the oxygen for only few seconds can cause complete inexcitability of some neurons. Okay. Next, effect of drugs. Caffeine, theophylline, theobromine, etc., which is present in the coffee, tea, etc., they will increase the neuronal excitability by reducing the threshold of the neurons. And most anesthetics, they will also increase the neuronal excitability. Okay. Sorry, most anesthetics, they will increase the threshold. So they will decrease the excitability and decrease the synaptic transmission. Next, divergence of the signals and convergence of signals. The divergence of signals, that is, you see here, a signal which is coming from here, they will be diverged in the same tract or it may be diverged in the multiple tracts. Whereas convergence of signals means multiple inputs, they will come to a single neuron. So multiple inputs, they will converge from, they will coming to a single neuron. Okay. So multiple source, convergence from multiple sources or convergence from single source. Then after discharge, that means that is a signal entering a pool causes prolonged output discharge called after discharge. It is lasting for few milliseconds to many minutes after the incoming signal is over. So if we have stimulated a nerve, now the stimulation is over, it is stopped, but the post synaptic that is neuron is still discharging its signals. That is the after discharge. Next, reverberatory circuit or oscillatory circuits. You see this circuit? This is the input, this is the output. Okay. And this output is further stimulating the input. So such circuits are caused by positive feedbacks okay, within the neuronal circuits. So this is a positive feedback system. Okay, so once stimulated, the circuit may discharge repeatedly for a long time. It may involve only a single neuron. The output neuron simply sends a collateral, okay, to collateral fiber back to its own dendrites or soma to re-stimulate itself. So it may send, okay, to re-stimulate itself, okay, or it may stimulate the input neuron. So see here, sometimes few additional neurons may be present in the feedback circuits. Those may be facilitatory or inhibitory neurons. Then the next video is about neurotransmitters. So in this video we have seen uh, the synapse, types of synapse, process of chemical synaptic transmission, then inhibition of the synapses, then the 14 properties of synaptic transmission. Next video is about neurotransmitters.